So, in the last session, I talked almost exclusively about DNA. Now let's have a look at RNA. Now, obviously, there are many similarities and a few differences. The similarities, once again, we've got a nucleobase joined via an N-glycosidic bond to an alcohol, in this case not deoxyribose, but ribose. And then these nucleosides form a polymer or polycondensate in which the individual building blocks, that is the connection of the base and the sugar, are connected once again via phosphodiester bonds from the 3' hydroxy group of one of the pentose to the 5' hydroxy group of the next one. Now, another difference, apart from a slightly different sugar being used in the RNA, is of course that in the pyrimidines we do have cytosine and uracil, not cytosine and thymine as in the DNA. And the major structural difference is that RNA in general is present in a single-stranded form, not as the double-stranded helix we know from the DNA. There are several forms of RNA, most of which I presume you're familiar with already from cell biology. So, on the one hand, there is the transfer RNA, in brief the tRNA, relatively small molecules having the job of transporting the amino acids to the site where these amino acids then are being connected to form one or the other protein. Then there's the ribosomal RNA, that's rRNA, which can be present in different sizes, and here the ribosomal RNA combines with the proteins to form the ribosomes, where the protein synthesis actually takes place. So ribosomal RNA and transfer RNA actually do work together, but they're not the same thing. When we're talking about the synthesis of the proteins at the ribosomes, the messenger RNA, the mRNA, once again variable in size, plays a role, basically having the job of pushing the amino acid sequences, pushing the amino acids and directing the sequence in the formation of the proteins. And there are further forms of RNA, most of which, once again, are far more important with respect to the cell biology than the biochemistry. So let's not go through the rest of this slide, but instead continue to see how DNA and RNA actually work together. So, you of course know that the DNA basically contains the building plan, so that the DNA actually fulfills at least two jobs. Job number one is this building plan has to be present in basically all the cells containing a nucleus. Of course, nucleus in the biological sense, not in the chemical sense. We're not talking about protons and neutrons here. And so, as this plan has to be basically ubiquitous, present in all the cells having a nucleus, the DNA has to be replicated, duplicated, multiplicated. This, of course, and I think once again you know that already from cell biology, happens via semi-conservative replication, because due to this base pairing, AT, GC, etc., basically every DNA brings along its own backup copy. Each individual strand of the DNA contains all the information required. Let's not talk about any mutations or any mistakes in the replication. This once again is a topic far more for cell biology than for biochemistry. So the duplication then works as follows, very, very simply put. Like I said, I presume you're familiar with it from cell biology. The double-stranded DNA is split, and then for each of the single strand parts, it's not completely split, it's just split at the part where the replication is supposed to take place. The synthesis of the counter strand takes place, which is simple for the strand we now look at from the 3' to the 5' ending, because then the synthesis, which obviously goes in the same direction but with inverse sign, so to say, from 5' to 3'. This is the 3' to 5' ending, so the counter strand would then be synthesized 5' to 3', one step after the other, a fully linear synthesis, and in the end you end up with a double strand. One of the two strands stemmed from the original DNA, which was replicated. The other is a copy, but once the copy is finished, it's not distinguishable from the original anymore. Now, if we take the 5' to 3' strand, the other one of the first split DNA, synthesis is slightly more complicated because the synthesis of the new strand has to go from the 5' to the 3' ending, which does not work if we have our original strand 5' to 3' here. So the synthesis works via Okazaki fragments, meaning that basically a part of the DNA is being read off and copied and then being connected with another part and another part and another part. So here there are several larger fragments which have to be connected once these individual parts were synthesized along the direction of 5' to 3'. That's not the topic I want to talk about right now, because this really is predominantly cell biology. Far more interesting, including on the chemical level, of course, is that the information of the nucleotide sequence of the DNA first has to be transcribed via RNA synthesis, where the second strand of the double helix, so to say, is being synthesized not with the building blocks of DNA, but of RNA. That is, using ribose, not deoxyribose, and using the respective bases used in the RNA. So it's the DNA strand determining the order of the bases of the RNA strand, and this transcription from DNA to RNA, and then working along the resulting RNA strand, where the ribosomes actually take the code of the code of the information stored in the RNA, which is the information stored in the DNA, to synthesize the proteins, that is, synthesizing the primary structure of the protein to be synthesized here by following the code of DNA/RNA to know which amino acids are to be connected with one another, so that you end up with first the peptide strand in the primary structure, and then the protein goes into the natural folding of the three-dimensional tertiary structure, or maybe even quaternary structure if we're talking about an oligomeric protein. All right. So we've got the transcription step and we've got the translation step. Let's have a slightly closer look at what transcription and translation mean. And once again, it is possible that I'm boring you till you're blue in the face, because you've all heard that in cell biology or anywhere else, but still it is also part of biochemistry, and occasionally repeating things might make it slightly clearer. So if we take the terms transcription and translation, we have to first have a look at what they actually mean. Transcription means we've got one set of symbols, one information stored in one form of symbols, which is switched to another form of symbols, whereas the translation then means transferring the meaning of the symbols. Now let's do this linguistically. If you look at this, series of letters, ktapt, what's that supposed to mean? Well, first you have to know what kind of symbols are we talking about. And even though they look extremely like Latin letters, they are not. Actually, they're Cyrillic. So some of the Cyrillic letters look exactly like Latin letters and have the same meaning, that is, stand for the same letter. The T-like looking thingy in Cyrillic actually is a T. The A-like looking thingy is an A. So we've got a T, an A, and another T here, but neither the C nor the P really stand for C and P, because this is not, those are not Latin letters. This thingy looking like the Latin C is the Cyrillic S, and this thingy looking like the Latin P is the Cyrillic R. In other words, if we transcribe, and that's exactly what transcription means, if we transcribe these Cyrillic letters to Latin letters from ktapt, we come to start. Yes, if you find these letters here, these Cyrillic letters in this order, they well, basically stand for the letters S-T-A-R-T. Whether or not this word now makes sense to you depends on whether you know the language, whether whether you know the language, that is Russian. Do you know that this word, what this word means? This is just what it would sound like to pronounce these Cyrillic letters. Now let's translate this. Okay, start. Let's translate this to different languages. Well, in English it's to begin or to start. In German, anfangen. In Chinese, 开始. But it all means the same thing, to begin something. Admittedly, this is slightly easier to do in English because you already see the word start and probably know what it means. In German, you would have to actually transfer to English first, but that's another thing. So, transcription, one code of information transferred to another code, like from Cyrillic to Latin, and then translation, one language, we're still talking about Russian here, to any other language you would like. So, for biology or biochemistry, the transcription is the step from DNA, one set of code letters, to RNA, another set of code letters. It's just different lettering. And the translation is from still working in the nucleobases or with the nucleobases attached to one or the other kind of sugar that really doesn't enter into it because the sugar does not bear the information. The information is stored in the bases, to protein. And for that, of course, you probably already know the base triplets in the DNA code, which stand for precisely defined amino acids, all of our proteinogenic amino acids. Now, this is the codon wheel, in German Codesonne. And if you read it from 5' to 3', as you're supposed to read it, just like you're supposed to read the primary structure of a protein from the N-terminus to the C-terminus, both DNA and RNA are to be read from the 5' to the start to the 3' ending, then you, for instance, see that the combination GAU stands for ASP, so aspartate, and GA and A stands for glutamate. Admittedly, some of the codes have more than one meaning. The AUG standing for MET, you know, methionine, is also a start codon, but then we're already back at hello cell biology again. And there are several codes, all of which starting with U, and then UGG or UGA or UAG, UAA. Those stand for, did I say UGG? UGG stands for tryptophan, I'm sorry. UGA, UAG, and UAA all stand also for stop. Okay, let's say it again. Now, we're still talking about our 20 proteinogenic amino acids, but by now you've seen that other amino acids also can and do play a vital role in biochemistry. Now, if there is any amino acid popping up anywhere in biochemistry, and they are not naturally occurring proteinogenic amino acids, then these amino acids must stem from somewhere. You've seen something like beta-alanine or whatever else it is. So there are two different possibilities how these amino acids come about. Either they are the result of something which is called post-translational modification, another term which brings lots of points in Scrabble, which only makes sense if you think about what post-translational modification means. If translation is the transfer from RNA to the amino acid chain, Then, of course, post-translational modification means changing something in the structure of the amino acids after the process of translation. So first, one or the other proteinogenic amino acid has been expressed here, has been synthesized according to the code, and then chemically modified. You might remember when we talked about the disease scurvy that hydroxylation of proline was extremely important, but hydroxyproline is not a proteinogenic amino acid, so this is a typical example of one naturally occurring amino acid not being proteinogenic, but being the result of post-translational modification. But by now you've seen several biochemical reactions, some of which even within one or the other biochemical pathway. But let's, for instance, remember the amination, transamination, where we take one biomolecule and then let it undergo one or the other kind of a reaction so that one functional group was exchanged for another one. So for instance, you might remember that we had an alpha-ketoglutarate which was reduced to glutamate by reducing the keto group of the alpha-ketoglutarate to formerly hydroxy group, and then exchanging this hydroxy group to an amino group. Actually, the mechanism is slightly different, but basically is we've got a carbon with a doubly bound oxygen attached to it. These two bonds are then in the process replaced by two single bonds, one going to a hydrogen, another one to the amino group. So it's an amination and the oxidation number of the respective carbon changed. So we're talking about reductive amination. The same thing works with aspartic acid, which can be formed via reductive amination from oxaloacetate, another biomolecule you've already encountered. I hope you do remember your gluconeogenesis, where a C3 body, the pyruvate, was first converted into the C4 body, oxaloacetate. So once again, you see how all these things are connected. Now, you might wonder where the ribose comes from. And of course, we know from the pentose phosphate pathway that ribose 5-phosphate popped up there. So this probably is one of the reasons why we've got so much of the ribose to be used in the RNA. But, and this is something, once again, going beyond not necessarily the scope of this lecture series. This is a bit of a question of how far we come in this lecture, this lecture series, this term, because, well, these are slightly different times than usually. But either at the end of this term or maybe somewhere later in an advanced biochemical course, you will find out that the ribose 5-phosphate is also the precursor for the formation of both the purine and the pyrimidine nucleotides. So once again, an individual molecule is being used for several completely different chemical reactions, which are so different that one might make the mistake of assuming that they're not connected in one way or the other. Now, for the next and final slide of this video session, after that we'll once again switch the topic, we'll return to our code wheel, but this time with some additional information attached to it. So here's our code wheel again, once again from 5' to 3' to be read. So there are all the codes you already know, and no, you don't have to memorize these the code wheel. If in the final exam a question regarding translation pops up, where perhaps the question comes from for which peptide does the following DNA code stand for which it stands, then you will definitely be handed out one or the other form of the code wheel. So you don't have to memorize that one. But let's please go on further. Of course, you see the various amino acids here, this time just for spatial reasons with the single letter code. And of course, you see that, for instance, UGU stands for cytosine. Cytosine, exactly what I didn't want to say. For cysteine, which of course is not to be mistaken for the cytosine from the one-letter code of DNA or RNA. So please know in which context you use which single-letter code. And then you will see that some of these amino acids here, which by the way are color-coded to tell you which are the basic ones, which are the polar ones, the non-polar ones, and which are the acidic ones. The acidic ones, of course, just the glutamate and the aspartate here. But there is also something else. You see this small yellow triangle here telling you that those are the positions where one or the other amino acid can relatively easily undergo one or the other post-translational modification. Now, it stands to reason that, for instance, the hydroxy group of serine, which amongst other codes is coded for by U, C, and then the whole third base, the third base really is irrelevant, all stand for the same amino acid, just U and C, as the first and the second in the code already determine what it is. So we're talking about a code which could be considered, just as you know it from general chemistry, where you call orbitals of the same energetic level, to be degenerate. Here we've got a degenerate code. You see whatever the third is stands for S. Some other codes are far more tightly knit. Look at how many base combinations stand, for instance, for tryptophan. Just one, UGG, that's it. There is no other code for tryptophan. Anyway, so it stands to reason that our serine and our tyrosine, having a hydroxy group, can, for instance, easily be phosphorylated. Because this is a typical thing happening, phosphoric acid forming an ester with one or the other alcohol, and whether or not you think of serine predominantly as an amino acid, it is also an alcohol, just as tyrosine is. Yet it is an aromatic alcohol, but it's still an alcohol. And of course there are other forms. We can, apart from phosphorylating it, we can methylate it. We can connect it with ubiquitin. You remember the ubiquitin from the redox equivalents and the like. We can exchange an oxygen by a nitrogen, then bearing a methyl group. Or we can use this alcohol function to be, for instance, connected with one or the other glycose, not glucose, glycose. I'm not telling you whether we're using glucose or mannose or, well, you know the important epimers of glucose, for instance. And of course, we can also have N-glycosylic bonds or N-glycosyl groups attached there. And if you look at the various amino acids, lysines, for instance, lysine with the epsilon amino group, here you see it's modified in at least 4 different ways here. If you see this SUMO here, now this stands for small ubiquitin-related modifier. This is used for several biochemical pathways, most of which actually go really beyond the scope of Biochemistry 101, the one you're listening to and watching right now. But they play a role for regulation of one or the other biochemical path. They play a role in apoptosis, which is as you know, the pre-programmed cell death, and many other things. Once again, this is not a slide to be memorized, but just to remind you that even though we're talking about a relatively large number of molecules, memorizing all of them is hard as it is, this is still a relatively small number of Lego bricks to play with, connecting them with other Lego bricks you're already familiar with. Yes, I know, I probably start getting on your nerves with my Lego association all the time, but biochemistry really is, in most cases, playing Lego at a slightly larger scale than in the general chemistry, but we're still just playing Lego. So please bear with me, we will continue playing Lego using already familiar bricks and some new ones when we come to the next topic. But this concludes the brief introduction to what can happen with nucleobases in connection with one or the other sugar, in connection with phosphate, and then of course secondary, tertiary and quaternary structures. This time not from the cell biologist's point of view, but strictly from the chemist's point of view. And chemists, like I already told you so and so many times, really enjoy playing Lego.